So good afternoon, everyone. Thank you for being here today. It's really a pleasure to welcome you to this first webinar of the ERDERA Training and Education Work Package series. Just a few words about this series. This initiative was developed within the ERDERA Work Package 20 in collaboration with different ERDERA partners. And the idea, so the aim of this series is really to address key topics relevant for the whole rare disease community and to address unmet training needs, which were identified last year through an internal survey. Today's webinar opens a dedicated series that will continue also over the coming months, so even after the summer break, and focus specifically on the ERDERA Diagnostic Research Workstream, introducing its objectives, organization, and ongoing activities. So it is now my pleasure to introduce the speaker of today's webinar, Professor Olm Gressner, who is the co-leader of the Workstream, coordinator of the European Reference Network for Rare Neurological Diseases, coordinator of the Solve-RD project, and also managing director of the Center for Rare Diseases at the University Hospital Tübingen in Germany. So before we start, just a quick reminder that all participants are muted, but you are, of course, free to post your questions in the chat during the presentation. We will address them later during the Q&A session at the end of the talk. And now, without any further delay, I'm very pleased to leave the floor to Professor Olm Gressner. Thank you.
Yeah, thanks, Magda. And thank you also for organizing the webinar series. It's a pleasure to contribute and a pleasure to present what we do in the clinical research workstream, the clinical research network, and the diagnostic research workstream. And I will be presenting now for 35-40 minutes, and then of course there's plenty of time for discussions and also questions you may have. Okay, so now you should be seeing my slides, all right? So as said, I'm presenting here the ERDERA Diagnostic Research Workstream, and it's an overview, so it's a beginning, and I'm the lead of this workstream within ERDERA. It's a beginning because also after me, not before summer, but after summer, there will be additional webinar presentations given by other members of the Diagnostic Research Workstream. So a second one will be on data sharing, data management, and ethics, and that will be given by Cornelia Elwanger and Leslie Matalonga. Then the third one will be on data analysis, reanalysis approach of the Diagnostic Research Workstream. That will be happening in October. Then the fourth one on long-read sequencing and optical genome mapping, which will be organized in December. Then next year, February, one on the result of the systematic reanalysis. The sixth one on first results from the innovative reanalysis approach, and by that you also already see a distinction we will be, and I will be coming back to, which is what we do as a standardized approach and as an innovative approach. Then 7, scaling up from distributed to federated reanalysis, and I'm also be touching on that later. And the last one planned as of yet is then on reanalysis of RNA sequencing data. And that's in June. So we are now on June 26. The last one will be in June 27, meaning that you will be having the opportunity to attend 2 webinars on the Diagnostic Research Workstream, basically throughout the year. Okay, so let me start with where we come from, and we come from Solve-RD in terms of diagnostic research, but also more concretely what we have been developing and establishing in terms of diagnostic research reanalysis. So SOFA-D, and Magda mentioned that I was also the coordinator of SOFA-D at that time, together with Olaf Ries. So SOFA-D is a research program which was also funded by the European Union. It concluded a bit more than 2 years ago. And SOFA-D, as the title already indicates too, was focused on solving unsolved rare diseases. And on the left-hand side of the slide, you see the patient journey through diagnosis, which was developed by ERDERA within SOFA-D to really represent, to depict what journey a patient without a diagnosis have to go through to, in the end, at least for about 50%, gain a diagnosis, and also for the other 50%, not yet receive a diagnosis. So they are then, at least to a certain extent, still in this diagnostic journey, and for them, and maybe also for ones which receive a diagnosis, although late, they really run then through a diagnostic odyssey. So SOFA-D is a research, or was a research program. We focused on patients with suspected genetic rare diseases and without a molecular proven genetic diagnosis. And we defined basically, or two basic inclusion criteria. So one was we really focused on patients or families for whom exome sequencing had been done already. So we did not start with the research if exome sequencing was not done yet. And of course, for the transfer from the diagnostic realm to research, informed patient consent needed to be in place so that we could then work with the respective data. And data means in this context phenotypic data, patient data, but also, of course, omics data, and to a certain extent also metadata. One of the very key principles we used with in SOFA-D, and that also carries over to what we now do in ERDERA, is that we base the work on the European reference networks. When we started with SOFA-D, the European reference networks were pretty young. So we started with SOFA-D in 2018, and the European reference networks were formed 2017. Which essentially meant that the journey, at least until the end of SOFA-D, of the European reference networks and of SOFA-D was kind of parallel. So we did not start with all of the European reference networks at once, but we started with those where we thought the respective expertise is in place. And also where we expected that the most gain can be achieved. So we started with 4. We started with the ones on neuromuscular diseases, then on rare neurological diseases, meaning basically rare movement disorders and rare cognitive disorders, then the one on rare congenital malformations and intellectual disability, and the fourth one was on is actually on rare genetic tumor risk syndromes. From the very beginning, we had also included undiagnosed disease programs, and that in a way epitomizes the move from the care system to research being done by SOFA-D. So the patients, the families which could diagnostically not be solved in the diagnostic setting, then were moved to research, and SOLVA-D was then the respective research program trying to solve additional patients, but also to find new rare diseases, of course, then also associated with additional diagnosis. Along the program, along the duration of SOLVA-D, we added two more buprenorphine networks. So we added the one on rare and complex epilepsies, and the one on rare immunological diseases, again with the same rationally. However, for us within SOLVA-D, this then represented a kind of what was an expansion, and also an expansion not only in terms of diseases being covered, but also in terms of sites being worked with. Essentially, at the end of SOLVA-D, the network we worked with was quite large, and was then, in terms of clinical expertise, in terms of clinical genetic expertise, also the foundation we carried over to ERDERA. And you see on the right-hand side of the slide the respective map. So in SOLVA-D, the main use case of the four we pursued was the one on reanalysis. So we basically collated unsolved exomes, partially also unsolved genomes, so from data from whole genome sequencing, and did actually organize a quite massive data sharing across Europe. We were able to collate more than 20,000 data sets. And if I say data set, this means that a data set is comprised of raw data, so exome, genome, raw data, plus phenotypic data, plus pedigree data. And those more than 20,000 data sets were then from approximately 30,000 families, by which you also see that we had, of course, data sets, more or data sets which belong to families, and in particular for de novo approaches, more than one family member is actually being needed to pursue those. So in addition to this reanalysis use case, we had three other use cases. One was on specific event cohorts. Here we actually tried to accommodate the respective disease needs by defining further analysis, basically by generating new omics data. Thirdly, we had a use case on ultra-rare diseases, and here we focused on very unique phenotypes and collated these very unique patients with specific phenotypic characteristics, and tried then by a phenotypic first approach to solve the respective identified patients and families, or included families and patients. And fourth, we had a use case which we called the unsolvables, and these were mainly syndromes on which many groups had been working on already, and we tried to solve those by using all the means we had at our disposal. So all omics data we could produce, all the expertise we could actually bring together. And at least to a certain extent, that was also successful. And I think it's also important to notice that what we started in SOLVA-D didn't stop. So even so the project is over, the effort, the organization, the data work is still continuing and actually contributing to solving further rare disease patients diagnostically. And we were able to publish quite a few papers, many of those in high impact journals, and we were also able to publish the first set on the unsolved cases, the reanalysis use case in Nature Medicine. So by a means of publication success, also here the project actually delivered. So looking now on what we actually did, and to explain a bit how Sofady developed its workflow, I would like to at least spend a few seconds on what is here now on this slide. So what we did started, of course, with data sharing. Then we did data harmonization, and that was actually more than necessary because we had that many sites from Europe. And you can imagine that having data coming from very many European sites also means that you have very many heterogeneous data standards, data formats being included into the program. So the first job was really to harmonize data. Then, based on the harmonized data, we did a comprehensive analysis. If you want a comprehensive reanalysis, and so we did a completely new variant calling. And then after this completely new variant calling, also so a standardized variant calling, also what we called, and I get back to that also later, what we called the data analysis task force, and the working groups of the data analysis task force added specific analysis types for certain variants. Then the interpretation was done by data interpretation task forces, so the experts, the clinical experts, being members of the data interpretation task forces, and together, so DETF and DATF, so data people and clinical people, if you want, we were able to solve at that time when we published it more than 500 families. And you see a bit the spread, how it was done. So we found in terms of variant types, so we found new genetic diagnosis in terms of SNPs and indels, but also more complex variant types. And you also see, at least as far as I can go into details here, the reasons why we were able to do so. So for some, it was just that new knowledge was available in terms of interpreting variants, which were called, and then we could use this knowledge. So we could, if you want, use the annotation of known variants from ClinVar, but we had also discovered completely new genes and completely new variants for which we needed the expert consensus, the expert collaboration to be successful. And in addition, and that's on the right-hand side of the slide, we were also able to use the ad hoc expert review on site. So once we were able to hand over the analysis results without using the collaboration framework of Sofady, the experts on site were also able to solve additional families. So you see, and that's my first figure here, and I'll give you a second in a sec. You see the distribution of the different variant types. So it's not only SNPs and indels. So actually, one of the success foundations of Sofady was actually that we, in addition to SNVs and indels, we also included more complex variant types, like combi number variations, like mitochondrial mutations, etc., etc. And this is actually also for us a basis why we were able to translate, to scale up what we did in Sofady to ERDERA. We saw that what we did in Sofady was bringing added value to all ERNs. So this is just the data, presenting the data from the first data freeze. And here you see that for the 4 ERNs which contributed to the the first data freeze, so RND, Ithaca, UNMD, and Genturus. At least if you combine new genetic diagnosis and candidate genetic diagnosis, then to a certain extent, equally gained from what we did in SOLVA-D. And actually, and I don't show you that here, also when you look at the country spread, so this is the ERN spread. Also, if you look at the country spread, you see that all of the countries actually benefit from what is being done in terms of research reanalysis. So there's no gradation in terms of where the patients come from, no matter where the patients come from, be it from the Netherlands, from Germany on the one hand, but also from Italy, Portugal, Turkey. They all benefit from a respective systematic reanalysis, research reanalysis approach. Okay, so how did we organize the work? We organized the work basically using two instances. One instance is the data analysis task force. This is the group of data analysts, bioinformaticians, and data scientists. And they organized the work according to variant types we focused on. And on the other hand, the second instance are the data interpretation task forces. And we had one data interpretation task force per ERN. And they were composed of clinicians and geneticists coming from the ERNs, contributing basically to the interpretation of the identified variants. And we have described that very early already, how we organized the work in a paper in the European Journal of Human Genetics, and that's here in Zurich et al. And those two, the data analysis task force working groups and the data interpretation task force, then defined what we called analysis projects based on the medical needs being defined by the ERNs, and of course, in terms of what is feasible, what can be analyzed, what data can be analyzed, and then what research can be produced, also informed by the respective working groups of the data analysis task force. Okay, so that's what we did in SOLVA-D. We were able to solve or get to an additional gain of more than 15%. And that's not the end. So we really see that based on the data being produced in SOLVA-D, but also based on the collaborations being established, more and more patients are being solved, so that the 15% might not be the end in terms of added diagnostic yield. So this is now the slide explaining the transition from SOLVA-D to ERDERA. So in SOLVA-D, we very much focused on whole exome data and whole genome sequencing data. We focused on short read data. We had, in the end, 6 ERNs contributing, forming data interpretation task forces. We had 23,000 data sets, and I explained what is being meant by data set. We had, even so, the country spread was well, at least for us when we started, quite large already. It was still a selection of EU countries, and we only used the distributed analysis type, so meaning that all the data we worked with was actually transferred. So distributed means that all the data, or the genomic data, or the phenotypic data, and also pedigree and metadata were actually being transferred to or uploaded, if you want, to one platform, which was then used to do the variant calling, and then based on the variant calling, we distributed further to 3 analysis centers, which were in Nijmegen, in Barcelona, and in Tübingen. So in ERDERA, and that's for us the next big step. That's really a scale up. We're now focusing, in addition to whole exome data and whole genome sequencing data, also on RNA sequencing data and epigenetic data, and in addition to short read data, also on long read data. So this is really omics type technology-wise a quantum leap we are doing here. Also in terms of disease groups, we are expanding, and the service we provide, the use case we implementing is now targeted towards all ERNs, and of the 24 ERNs, 23 are defined by disease groups. So to those we convey the invitation to collaborate, and more actually accept. If you know the ERNs, if you want, you can actually divide them at least to a certain extent into events focusing, or let's say having also or covering also genetically caused diseases in their disease portfolio, and there are also events which more focus on intervention, like complex surgery, and those of course then don't have that much of incentive to collaborate. But in the end, we want to collaborate with more than 15 events in what we do in ERDERA. The numbers, of course, will increase significantly. In terms of geographic outreach, we go beyond what we had in SOLVA-D. So it's now European Union and also including, of course, underrepresented countries. And we will be also developing the federated approach, meaning that we won't, for all the data we analyze, share the data, so upload the data at the platform, but also leave the data where they are and do then a harmonized analysis at the respective sites. Okay, so I guess you all have seen the House of ERDERA until now. So what we do belongs to the Clinical Research Network, and we have in the Clinical Research Network 3 work packages, one on diagnostic data availability, a second one on genome reanalysis research pipeline, and a third one on innovation to shorten time to rare disease diagnosis. And that's the schematic overview, and it goes from left to right. And if you want, back then from right to left, and it's actually kind of a circle being operating. It starts all with data availability because we need respective standardized data to be able to do what we do. Then in work package 7, we implement the reanalysis, and I explained the DETF and the DITF collaboration framework. And you see that here are already more events included than what we had included in SOLVA-D, and also the federated approach is being presented. And that's really the exciting thing. We introduce innovations as we go along. So we started with ERDERA, with the Diagnostic Research Works in ERDERA 1.5 years ago. And we're now introducing, in addition to exome sequencing data and whole genome sequencing data, also RNA sequencing data. So again, for the reanalysis, so based on data already existing and being used in the diagnostic setting, but not able to solve the respective patient diagnostically. And we're also using optical genome mapping data, and that's not that far away. We're also introducing long read data into our reanalysis research pipeline. Okay, so what I now do is really giving you a bit of an overview of what we cover in the work packages. So work package 6, and that's the one on data availability. So for us, it's really, and I think I made that clear, work package 6 is expanding diagnostic data availability and the respective collation effort. We need to implement data standardization and quality criteria for other omics data besides whole exome data and whole genome data, genome sequencing data. I explained that also as well. And of course, what we do doesn't stop with the data analysis, so result generation and interpretation, but we feed that back to the clinician, to the geneticist, and we want to also organize, and we have been doing that, a collaborative variant interpretation. So we have one governance body, which is called Data Availability Committee, in which the decision is being, the main decisions are being taken. And then here, I think importantly, which I will be showing a slide on as well later, we have a specific network on the underrepresented countries. And I explain in a sec a bit more on that. So because when you start working with data, and in particular with patient data and genomic data, you need to comply with the GDPR, so with the data protection requirements as being set up in Europe. So we have developed and implemented a data sharing framework. So that's in place already and is being used, and it's being based on joint controllership. So essentially, the build-up is that the three main data processors, which is Tübingen, Barcelona, GPAP, and Groningen, signed a first co-agreement, and then all other partners actually accede to the data sharing framework agreement. And we also have a separate data processing agreement with EGA, with the European instance of EGA, because that's just what they required to be used as a data broker. So basically for us to organize our data transfer. And as of yet, so at present means that the status in March, so we are a bit further now and have more centers that have joined the data sharing framework agreement as joint controller already. So we had 44, and now we are close to 50 already, and it's getting more and more in terms of partners joining the joint controllership, and only, and that's important, only if you join the data sharing framework agreement, then you're also able to contribute to the reanalysis research effort being done in ERDERA, and only then you can also access data. So this is really the legal environment, the legal framework we have been building. I mentioned data standardization, and I don't go into much detail here. I just mentioned that we have developed prospective data standards and quality criteria for all the data types we're working with, including long-read data and optical genome data, and that's of course now being used mainly in data in Workpack 6, so for data availability, but also beyond, because when we now implementing also federated analysis, this is of course the very basis to be able to, in the end, also share aggregated results file, because if you don't work with the same kind of data standard, then it won't work. Okay, so that's a long time ago, at least in terms of how quickly we process and how quickly we proceed and progress. So this is the a picture showing you where the data came from in the first year. So in the first year, we were able to share more than 10,000 whole exome, whole genome sequencing data. You see the countries with the respective numbers. This was the very beginning, but I think the baseline here is that we were able to start a massive data sharing effort much more quicker than we were able to do so in SOLVE-ID. And you see the much larger number for year 2. So using what we established being set up in year 1, we're now really able to scale up on what we did in SOLVE-ID. And that, of course, also benefits the underrepresented countries. And underrepresented here means no gradation in terms of economic development or so. It's really a gradation in terms of what we have seen as of yet in terms of contribution to research projects. And that's also a specific offer we make to the countries being called underrepresented countries, and because we want to, and specifically bring about benefits for them, we have formed that network, and we also see that at least for the organization of interpretation, this kind of network is quite useful. Okay, so submission, and I'm talking here the distributed approach, so not the federated one. For the distributed approach, 3 kinds of submissions are needed, and I touched on that. So it's phenotypic data, so clinical phenotype, it's metadata and raw genomic data, and it's all being submitted to the ERDERA GPUB in Barcelona. And then for the analysis, the data are being used first for variant calling, and then of course also for the respective data for the respective variant type specific analysis and interpretation. Okay, that was data availability. Now the pipeline, the genome reanalysis pipeline, and I don't need to explain a lot here because you have seen the figure already. But I'm showing the figure again because it also indicates that there's a continuation in terms of collaborative framework, collaborative ecosystem being used. So the collaborative ecosystem, which worked so well in Solvadi, we're now also using in ERDERA and using that also for the scale-up I have explained. So in terms of workflow, and that's if you want to contribute, and this is also an invitation, and I will be repeating this invitation after the presentation to contribute. The following steps will be happening. So it's first, of course, the submission of data of diagnostically unsolved patient. Then there's a central reannotation and reanalysis of the data. That's now being done by the newest Dragon pipeline, plus a few further variant calling tools. Then there's additional analysis tailored to variant types. That's being done by the DETF working groups, and by now you know what that means. Then we have case-based interpretation. So basically all submitting centers get back their result data, and they can have a look and find out as to whether they're able to solve their patients. For more complex interpretations, so for group patients, we use the ERN-led informed DITFs, data interpretation task forces. You also heard what that is. And then data will be made available for further analysis. So for the big spikes of data, large spikes of data through HEA. And of course, in addition to providing, at least after translation into the clinical setting, in addition to providing benefits, added value for patients, we also have scientific added value, which is produced by ERDERA in terms of publications and, of course, in terms of new knowledge being produced. And if you now look at the figure on the right-hand side, there you see 2 tiers being depicted. And we actually have also a 3rd tier, but here and now it's just, I think, important to understand that we have a standardized reanalysis, which is done for all data. And we have innovative reanalysis approaches, which are done for a subtype of the data, focused on specific variant types, focused on specific disease groups, etc. And this is then followed different routes, because when implementing actually the standardized approach or the systematic reanalysis, we follow strict timelines because we want to deliver to provide data and results to the submitters at clearly defined points in time. Okay, so this is a bit of repetition again, in particular in terms of showing the tier 1 and tier 2 reanalysis. And you see there's a bit of difference in terms of timelines being applied. And the one bit I want to mention here is that while for the tier 1 approach, for systematic reanalysis, all is being done via the ArdeConnect GPAP. So this is our main, well, if you want, basis platform we work with. It gets a bit more spread out. What is being done for the tier 2 and tier 3 analysis approaches, because that's then more on using innovative approaches and working with the data in innovative ways. But in the end, and that's important, whenever results are being produced, which might lead to new diagnosis, these data are being given to the submitting clinical centers, to genetic centers for interpretation, and then of course also are subject to group interpretation being done by the ERNs. Okay, so I mentioned that we don't stop with 4 or 6 ERNs in ERDERA, and here you see the full spectrum. And we have organized the large group of ERNs now contributing into 3 groups, which we call super DITFs. So we have one which combines Ithaca, the one on metabolic diseases, epicare, and the one on craniofacial malformations. That's super DITF1. Then we have a super DITF2 combining NMD, so neuromuscular diseases and rare neurologic diseases. And we have a super DITF for the rest. This is in terms of organization, just having people, having also experts being responsible for the interpretation taking place. Of course, and I think this is also a message I want to get across, we have DITF, so data interpretation task forces per ERN. So once we have respective data submissions of an ERN of a disease group which belongs to ERNs, we form a ERN DITF, and they then organize the data interpretation. Okay, this is also nothing I want to spend too much time on. So this is now the organization of the data analysis task force in ERDERA. So we have again working groups, and you see it's on small DNA variants, large DNA variants, integration and decision support, and statistical testing and association studies. And you also see at the bottom, linked with Work Package 8, already formed working groups of the data analysis task force for the new data types, so for long read sequencing, for optical genome mapping data, and also for RNA sequencing data. All right, and that's something really exciting, but also challenging and complex. So we know, at least from what we did and now what we have been doing in ERDERA and in SOLVE-ID, how the distributed approach works. Now we transitioning to the federated approach. So the intention is, and we actually are quite close to implementing the first real use cases. We want to implement also a federated approach, so meaning that the data stay where they are. But basically, if functional in respective national data infrastructures, and then the data analysis is being done there, the interpretation is being organized, and then either a federated reanalysis or federated interpretation, or also the data standardization and the standardization of methods being used for analysis is being done. And all this is also now being covered by ERDERA. Okay, so Work Package 8, innovation to shorten time to diagnosis. That's, as explained, focusing on cutting-edge genomics and integrating multi-omics approaches. I also explained that the key activities focus on the innovative omic data types, and they also mentioned that we have a specific interest in underrepresented countries. And here in Work Package 8, we combine both. So we combine the new data types we work with, and we see also that in terms of availability of those new data technologies, there's a well, let's say an implementation gradient. So it started in few countries like Germany, Netherlands, France, etc. We see that, and there are countries in which this implementation takes longer. So we're using ERDERA by means of Work Package 8 to start with research by giving access to the respective technologies by means of research, and we have use cases focusing on long-read sequencing, OGM, but also RNA sequencing in underrepresented countries. Well, this is essentially what I just explained. So we use long-read data, long-read genome sequencing, and optical genome mapping. And this is just the first use case, the initial implementation. That is also an invitation. So the invitation, they have two invitations. One is to contribute to the reanalysis effort, but the second invitation is also to contribute to the use case implemented in the underrepresented countries for the new types of data. So if you have an interest, please, by all means, get in touch with us. All right, so of course, there are plenty of documents, resources already available, and we have put most of those on the ERDERA website, or will be doing so. And the links when the presentation will become available are then available, but also, of course, if you're interested in getting access to one or another resource, please, by all means, get in touch with us. You see our functional email addresses under the please contact us phrase. So please, by all means, use the functional email address to get in touch with us if you want to contribute, if you want to collaborate, but also if you have questions. So I end my presentations by just indicating, pointing to the next webinar being given. That's a bit time, so it's after the summer. And then what I just touched on, the data sharing, the organization of the data sharing, the data management, but also the ethical aspect will be explained in detail, and that will be done by my colleagues, Cornelia Ellwanger from Tübingen, and by Leslie Matalonga from Barcelona. And with that, I do thank you for your attention, and I'm happy to hand over now to the organizing team. Thank you.
Thank you, Holm, for your presentation. It was very interesting indeed, and I'm sure there are many questions awaiting us. We do have the Q&R functionality in this webinar, which is why I would ask you to put in your questions there, so Holm can reply to those in a moment. But before we continue with your questions, we were thinking of doing a short Q&R ourselves with you as participants. And so there are 5 questions that we would like to raise now, and I'll read one after the other and put them into the Q&R section. I hope you can see the first of them. It's the question is the ERDERA Diagnostic Research Workstream a direct successor to Solve-RD project, building on its infrastructure and expertise? So if you have a reply to this question, it would be great if you could reply in the comment to this question.
And it's a simple yes or no answer you can provide.
It's not complicated indeed. Let's Maybe Holm, you would like to answer the first question.
Yeah, it's a clear yes. I mean, make that very clear, I hope.
Okay. Our second question to you is participation. Just a second. I'm opening it. Is participation in the joint controllership of the diagnostic research workstream restricted to ERDERA beneficiaries only? If you want to reply to this, there is the possibility to put in a comment.
And this one is a bit more tricky because I didn't not mention it in particular. Maybe it was clear from the things I said, and I can comment on it once we have given a bit of time.
Yeah. So Holm, if you would like to answer to the question.
Yeah, so here the answer is no. The very condition to contribute is that a institution joins the joint controllership of the data sharing framework agreement, and it doesn't matter as to whether this institution is a ERDERA beneficiary or not. So also non-ERDERA beneficiaries can collaborate, contribute into the ERDERA diagnostic research workstream.
So we have a third question also that I would like to submit now is are data submission standards for sequencing modalities beyond VES and SRVGS currently being developed or adopted within Work Package 6? I'm pasting the question in the Q&A. If you would like to have a look, you can reply within this functionality as a comment as well. Maybe Holm, you would like to answer. I believe the Q&A is a bit challenging to.
I mean it's yeah. So it's a clear yes, as explained. We started with whole exome data, sequencing data, and whole genome sequencing data, and in Solver-D, where we mainly focused on whole exome sequencing data. And now we include also RNA sequencing data, optical genome mapping data, and long-read sequencing data. So it's, at least in terms of what is available, a full portfolio, if you want.
So we have the fourth question out of the 5, which is the reanalysis of unsolved VGS datasets in VP7 in Work Package 7 carried out exclusively by the submitting centers without any central pipeline? And again, you have the possibility to reply in the comments part to that question. Again, Holm, if you would like to reply to the question.
Yeah, so it's a bit of yes and no. So at the moment it's yes, because we mainly implementing the distributed approach, and for all the data sets we have been collated, will be collating, a centralized reanalysis pipeline is being used. Once we come to the federated approach, and that's also the answer to Pipis, that might be different. So then I think data might not be needed to upload it to the central platform we work with, but then the pipelines could also be implemented where the data are, and that will hopefully be the national genomic infrastructures. But for now, it's a yes.
Okay. So we have our final question, which is just a second, I'm reading it out loud. Does Work Package 8 offer centers from underrepresented countries the opportunity to have their samples processed using advanced technologies? or OGM, even if those technologies are not available locally. I think it's best if you reply home, because I I fear that the interaction part with the Q&A is
yeah. I'm already reading the questions in the Q&A chat. Can you briefly repeat the question, because I was a bit distracted by one of the questions?
Yeah. So does Work Package 8 offer centers from underrepresented countries the opportunity to have their samples processed using advanced technologies?
Yes. I think that's an offer I explained, and that's an offer which stands and also will be implemented as we go along. So if you are from an underrepresented countries, if you are interested to contribute, please get in touch with us.
Thank you. Thank you very much. So I think we have also a couple of questions already in the chat. We have also Magda chairing today. So if you want to join us, Magda, to manage a little bit the questions, just before I hand over to you, I would also like to share with you our survey. So it would be great if you could fill in the survey after our webinar, and sharing it here in the Q&A section as well, just for you to have a look at it later on. Thank you. I'm handing over to you, Magda.
Thank you, Eileen. Yes, indeed, there are a few questions in the Q&A section. So there was a first question from Vanda. You already answered on, but I think it is worth, making anyone aware. So the question was, can one contribute with samples within research activities of a GTC 2026 project? I don't know if you want to.
Yeah, and I definitely want to answer that again. So the thing is that the GTC 26 call is not yet finalized. So the first phase application, which have qualified for the second phase, are now preparing the second phase applications. If you're successful, one of the conditions you get funding is that you contribute and collaborate with the Diagnostic Research Workstream within ERDERA. So it's a clear yes. So we expect that when you have unsolved patients, that you contribute, collaborate with what we do in ERDERA. So yes.
Thank you. And the question related to this one, can we then include a direct collaboration with the DITF and DITF and DETF in our project to strengthen the research activities and RD workflow alignment?
Yeah, if that's existing already, of course, I think you should. If you have, and I understand that the question also relating to an application, if the application is actually on a disease group for which a DITF, so a data interpretation task force, already exists, I believe that would really strengthen the application, because it will then already in the ecosystem will be using and have been using and are using in the Diagnostic Research Workstream.
Okay, next question. I have a question. Applying to a grant, we have a group of 100 patients with a genetic diagnosis. We will test them functionally. If at the end of our project we have patients for which we could not prove effect on the function, do we have to submit this patient?
Not fully understand the question. So if the diagnosis is established already, and that's how I understand the question, then these are patients samples with the diagnosis already. Yeah. And I think then we can contribute only little. So the thing what we really focus is diagnostic solving undiagnosed, unsolved patients. So we don't focus on functional analysis. I think it might be a bit different when we're talking the existing of RNA sequencing data, which might be useful for respective functional interpretation. But I think that needs to be discussed in detail. So I can't really give a detailed answer on that one.
Okay. So another question, thank you for the great presentation. This is an important effort. The transition to a federated pipeline for small sites seemed to happen only towards the end of the project, 2031. Is this correct?
So it's a multi-layer approach we're following as regards the federated approach. So we start from the highest level is actually the discussions we have with the genome data infrastructure and the 1+1,000,000 genomes project, because that's what defines actually, at least on European level, the legal, ethical, and data governance framework. Then the second level is the discussion with the national sites. So there are countries in which there are already functional national genomic data infrastructures, and those are, of course, the ones which we would like to collaborate with, because then a respective national effort is already underway. If that's not yet functional, not yet achieved, then we're also working with sites. This is the third level, and we also have been discussing with sites on implementing the federated approach. So I think it depends a bit on timelines, on size of the site we're talking. So I would say get back to us and start a discussion. Then we can do a shared assessment, a joint assessment as to if, and also to what extent it might be sensible to join the federated approach we have been pursuing now.
We have in the chat a follow-up on the previous question. So following up on my question regarding functional diagnosis. Diagnosis for this patient is not established. Variant are VUS. Or do you consider them diagnosed if two variants are identified?
No, then I think it's then the answer is easier. Then for the ones you're not able to establish the diagnosis, I would recommend to include them into the diagnostic research workstream.
Another question you already addressed. I don't know if you want to say more. Thank you all. This is a great resource with the ongoing work. In the current ERDERA call for solving unsolved cases, is it mandatory to submit our unsolved omics data to a diagnostic research workstream?
Yeah, it's a clear yes. So that's what we because we want to collaborate with the HANA GTC project, and we want to also make sure that they benefit from what we have set up. So it's according to the call text, it's a yes.
Other question, the development of pipelines for long reads, omics and RNA-seq seems very timely. If projects within other ERDERA funding efforts are also working along these lines, are there collaboration opportunities or even requirements?
So it's yeah. So it's a yes and yes. So there is of course I showed the House of ERDERA, and I didn't explain much of the other parts which belong to the ERDERA house. What I just explained is one of the parts of the ERDERA house. But of course, there are also other parts like the Data Service Hub, and also within the Data Service Hub, respective methodologies are being developed, and we collaborate, and we are eager to collaborate. So I would say, it's not obligatory, but we do it because it's beneficial in terms of what we can do. So I think it's a single yes to this answer.
Thank you, Olma. Last question so far. Concerning data sharing agreement, are there guidelines for the participants to simplify and standardize the obtention of a local ethical agreement for sharing data internationally, as this can be restricted in certain countries?
Yeah, that's also the benefit of SOFRD. So we have a number of ethical protocols that were submitted for respective data sharing and that work, which are also, of course, willing to share. that can be used for the respective national or local submissions. As of yet, and that's of course the heterogeneous landscape we work with in Europe, there's, at least as far as I know, only one country in which a country submission of its ethical protocol is possible. All other countries have site-specific local ethical committees. So we are willing to share whatever resource we have, but in the end, each site needs ethical permission from their respective ethical committee, and that's what needs to be there. Or if you have that in place already, I think the consent the patient has signed should be covering the respective data sharing.
Thank you all. There are no more questions. I think we can close the webinar. So thank you all for this insightful presentation, and thank you all the participants for joining us today. So do not forget to fill the survey Eileen posted in the chat, and we hope to see you again at the next webinars of the Diagnostic Research Workstream. So the next one will be in September on data sharing, data management, and ethics. Thank you all, and have a nice day.
Yeah, thank you.
Thank you. Bye-bye.